viewers, welcome to this particular lecture on photosynthesis. In this particular lecture, I shall be discussing different methods of carbon fixation except the common method, which is the C3 cycle or the Kelvin cycle. And this method include the C1 cycle, which is exhibited by different bacteria and archaea, the C2 cycle, which is the photorespiratory process, which normally occurs in plant exposed to high sunlight and high concentration of oxygen, the C4 cycle, which is exhibited by some specific tropical grasses, and the crassulacean acid metabolism or the CAM pathway, which is exhibited by different crassulacean plants or the succulents. Now, the objectives of this particular lecture include to understand the mechanism of C1 pathway, to understand the mechanism of C2 pathway, of course the photorespiratory pathway, and also to understand the process of C4 cycle, and to know the mechanism of crassulacean acid metabolism. I shall start with the C1 pathway. It is so called because the first photosynthetic product is a C1 product. By fixing one molecule of carbon dioxide, they produce a C1 compound, which is mainly methanogenic compound. And as the name signifies, it is exhibited by different methanogenic bacteria. One of them is Aquifex aeolicus, and this particular bacteria is also called extremophilic organism because it is growing in a hot spring where the temperature might go up to 90 degrees centigrade. Now, the process involves a unique pathway. Basically, it is fixing of carbon dioxide and production of some methanogenic compound. So, this process is like this: carbon dioxide, when it is fixed, it is producing methanofuran or MFR, which is gradually converted to H4-MPT, or it is a derivative of methane called tetrahydromethanopterine, and gradually this will be converted, or it will be binding with another compound, that is the tetrahydrofolate. Finally, it will be converted to methionine and it will produce amino acids like serine or glycine. This entire process, previously it was called only a methanogenic process, so it produced methane. But now it is understood that methane is producing some intermediate compounds or protein, which ultimately will be converted to amino acid. So, it is a very interesting pathway, and it is also called the Wundt-Jungdahl pathway. And basically, it is also correlated in plant where there is normal glycolytic pathway or gluconeogenesis pathway producing keto acids. Now, let us consider the second process, which is the C2 pathway. It is so called because the first photosynthetic product is a C2 compound, and that is called phosphoglycolic acid or phosphoglycolate. What happens is RUBP, the ribulose biphosphate, is actually absorbing oxygen along with CO2, and that is why it is producing phosphoglycerate, which is a 3-carbon compound. Along with that, it is also producing a 2-carbon compound phosphoglycolate. And because of that, the entire process of photosynthesis C3 pathway gets delayed. If we try to understand the process involved in the process, it is like this. Basically, the oxygen and carbon dioxide is taken up together by the compound, which is the Rubisco or RUDP, and the Rubisco is the enzyme which will ultimately produce the glycolate, and this glycolate, through specific connecting molecule or connecting receptor, it will be getting into peroxisome, and this peroxisome will convert glycolate to glyoxylate, and this glyoxylate will produce hydrogen peroxide. Finally, there will be some conversion of glyoxylate to glycine by taking in amino group, and this particular amino transferase will convert glyoxylate to glycine, and the glycine is a structural amino acid which will be getting into the mitochondria, and there the glycine might be converted to other amino acid like serine, it might take up another amino group and that will be producing serine and it will be utilizing NAD, and this serine again comes back to the peroxisome and this serine will be converted to hydroxypyruvate by losing the amino group, and this hydroxypyruvate will be reduced again by NADH linked reductase to produce glycerate, and finally this glycerate will be converted to phosphoglyceric acid, and this phosphoglyceric acid will carry on the normal C3 pathway. So what we actually see, the entire process which is a normal photosynthetic C3 pathway is getting delayed. So we might think that this is a very much boastful process, but actually it is not an entirely boastful process because there are some utility molecules produced, which includes the structural amino acids like glycine and serine. Though it is producing hydrogen peroxide, but because of the activity of the peroxidase enzyme, this hydrogen peroxide is broken down and oxygen is produced. And finally, even if it is delayed, the process also liberates PGA and the normal C3 process is carried out. But as I told you, this particular process will only be observed at certain times, and that include when the oxygen concentration is relatively high than the carbon dioxide concentration, and that is because of this fact that the enzyme Rubisco, which will try to fix both oxygen and CO2, and it will not be dedicated only to CO2. Furthermore, high amount of light intensity actually increases the temperature. So high temperature is also an useful point to be noted, which will carry out photorespiration or C2 pathway. Now, C2 pathway, as I told you, can be operated by any plant. Any photosynthetic plant will exhibit C2 pathway, but some plants actually will evade the photorespiratory pathway. And naturally, these plants will have more photosynthetic productivity because the carbon dioxide concentration is high. And this includes the C4 plant. They are so called because they produce the first photosynthetic product of oxaloacetic acid. But the C4 plant, because of the activity of PEP carboxylase enzyme, is producing excess carbon dioxide inside. It is also evaded by the CAM plants or crassulacean plants, where the photosynthesis actually is operating during night because the stomata is open during night. Carbon fixation occurs at that time, it produces malic acid. So that is called night acidification, and this malic acid during daytime when the stomata is closed is producing excess of CO2. So when the excess of CO2 is present inside the plant body, naturally there will be normal photosynthetic C3 pathway and there is no possibility of the photorespiratory pathway to carry out. It is also exhibited by certain algae, the green algae like Chloromonas, and this is basically carbon concentration mechanism. This carbon concentration mechanism is carried out mainly because of the shuttling of carbon dioxide into the particular photosynthetic apparatus where the carbon fixation will be carried out. So this CCM mechanism is very much operative near the Rubisco enzyme, as a result of which Rubisco always gets high concentration of carbon dioxide and there will be no formation of C2 molecule. It is also exhibited by hornwort or anthoceros, where they also have this carbon concentration mechanism in the pyrenoid, and this pyrenoid or in the chloroplast, carbon dioxide in excess will prevent the oxygen fixation by Rubisco enzyme. It is also exhibited by different cyanobacteria. Now this cyanobacteria is having different types of carbon fixation receptor, and they are the NDH13, NDH14, and in freshwater cyanobacteria, in addition to this NDH13 and 4, there will be BICA1 and SBTA1 and BCT1, which all actually will allow the fixation of bicarbonate, and so much high concentration of bicarbonate actually is occurring due to more sodium ion influx. So it is basically a co-transport of bicarbonate and sodium, but the result is there will be excess of bicarbonate available inside the cell, which will allow the Rubisco enzyme to fix carbon dioxide, and there is no possibility of having photorespiration. But in alpha cyanobacteria, there will be some smaller molecule receptor like NDH14 or SBTA2 and BICA2, which will also have this bicarbonate, and there will be this fixation of bicarbonate or carbon dioxide by Rubisco, as a result of which the cyanobacteria also evade the normal photorespiratory pathway or the C2 cycle. As I told you, photorespiration is important for the plant itself. That is, it may be necessary for the assimilation of nitrate from the soil. Several physiological processes may be responsible for linking photorespiration and nitrogen assimilation, and it will increase the availability of NADH or the conversion of nitrate to nitrite. Of course, it will also influence wide range of process that include the photosystem 2 functioning, and it is the main source of hydrogen peroxide, and as I told you, the hydrogen peroxide can itself be metabolized within the plant. It will also regulate plant hormonal activity, growth and development, and most importantly, it will result in programmed cell death or apoptosis in worn out cell. And it is postulated that this photorespiration is actually acting as a safety valve against free radical damage. Now let us consider the next important carbon fixation pathway, and that is the C4 pathway. This particular process is so called because the first photosynthetic product is oxaloacetic acid, which is a C4 compound, and it was also called Hatch-Slack pathway because it was denoted by Hatch and Slack way back in the 80s. And this particular process include the sugarcane, which is a tropical plant, and also the succulent plant like cactus. But these plants in general have a unique anatomy, and this anatomy is they are having the vascular bundle surrounded by a unique bundle sheath, and thereby the mesophyll tissue and the bundle sheath both are responsible for carrying out the photosynthetic process. So we have two types of photosynthesis here. One is the mesophyll photosynthesis, another one is the bundle sheath photosynthesis. To be specific, it is carbon fixation. Technically, this particular C4 pathway can be classified into three different types, and they are the NADP-ME type, NAD-ME type, and the PEPCK type. So there are three major types, and these three major types are mostly formed because of the unique enzyme. Let us consider the first type, that is the NADP-ME type, where the carbon dioxide is fixed and the PEP carboxylase is active, and that will produce the oxaloacetic acid in the bundle sheath. And this oxaloacetic acid will be converted to malic acid because of the reducing process of the NADPH, and this malic acid will be getting into the inner part, that is the mesophyll, where they will be converted to pyruvate, and this particular enzyme is NADP-ME, and that's why it is called the NADP malic enzyme, and the process is NADP-ME, and that will result in carbon release or decarboxylation. So there are more carbon dioxide generated inside the mesophyll, and it will produce pyruvic acid, and this pyruvic acid again comes back and will be converted by phosphorylation to phosphoenol pyruvate, and the cycle continues. The second important process is called the NAD-ME process, and in this case carbon dioxide is fixed, it is converted to phosphoenol pyruvate, carboxylase enzyme is converting it to oxaloacetic acid, and the oxaloacetic acid will be getting the amino group from glutamate to produce aspartate, and this aspartate will get into the mesophyll tissue, and the aspartate amino transferase will again reconvert it to oxaloacetic acid, and this oxaloacetic acid will be converted to malic acid under the action of the NAD-ME enzyme or the NAD malic enzyme, and thereby NAD is produced, and this particular malic acid finally will be reduced also by this NAD-ME enzyme to pyruvate, and this pyruvate will be converted to alanine by getting the amino group from other amino acid, that is amino transferase. But in the process, while generating pyruvate, it will produce carbon dioxide that will be operating the Calvin cycle, and this alanine which is produced inside the mesophyll will be getting back to the bundle sheath and will be converted to pyruvate by losing the amino group to due to the process of aminotransferase, and the process continues. The third important process is the PEPCK type C4 cycle. It is so called because the enzyme is phosphoenol pyruvate carboxykinase. In this particular process, what happens is carbon dioxide initially is fixed by phosphoenol pyruvate carboxylase, and it will be converting phosphoenol pyruvate to oxaloacetic acid like in the previous process, and this oxaloacetic acid is converted to malic acid via the dehydrogenase or malate dehydrogenase. This malic acid will get into the mesophyll tissue and it will be acted upon by NAD-ME enzyme, and it will be converted to pyruvic acid. Actually, at that point of time it will generate carbon dioxide, and this pyruvic acid again will be converted by aminotransferase to alanine, and this alanine again will lose the amino group and come back to the pyruvate, and in the bundle sheath cell this pyruvate again will be undergoing phosphorylation to produce phosphoenol pyruvate. But why it is PEPCK? Because a part of this oxaloacetic acid which is produced by phosphoenol pyruvate is getting an amino group that is from aspartate aminotransferase, and this enzyme will convert oxaloacetic acid to aspartic acid, and this aspartic acid will be converted to mesophyll, where again it will be reconverted to oxaloacetic acid by aminotransferase, and this oxaloacetic acid by the PEP carboxykinase will be converting the oxaloacetic to phosphoenol pyruvate, and here it will generate carbon dioxide with the help of ATP. So what happens is inside the mesophyll there is excess of carbon dioxide produced, as a result of which the Kelvin cycle becomes highly efficient, and this is for this reason that this excess of carbon dioxide being fixed, the photosynthetic productivity of the plant increases, and that is also another reason the PEPCK gene from plants or wild grasses which exhibit the C4 cycle may be converted to rice to produce more photosynthetic rice, which is in colloquial term called Pepsi rice. Now we come to the last important process of carbon fixation, which is the crassulacean acid metabolism. In this particular process, actually the first photosynthetic product is an acid which is the organic acid or malic acid. It was denoted way back in 1804 by Saussure, and this process actually is exhibited by plants belonging to Crassulaceae which shows negative circadian rhythm. That is, the stomata, they remain active or open during night and they are closed during daytime. What happens is during night time, when the stomata is open, the carbon dioxide gets in and that will be converted to oxaloacetic acid, or basically this carbon dioxide or the bicarbonate will be utilized by phosphoenol pyruvate to produce oxaloacetic acid, and the malate dehydrogenase will convert the oxaloacetic acid to malic acid, and this malic acid will be stored inside the vacuole. It remains inside the vacuole, and then during daytime what happens is this malic acid again will come out of the vacuole, and by the malic enzyme or malic dehydrogenase or malic reductase it will be converted to pyruvate, and this pyruvate will be getting the phosphate from ATP converted to phosphoenol pyruvate, and this phosphoenol pyruvate under the action of PEP carboxykinase again will give oxaloacetic acid, and that will be converted to malate again, and this way the cycle continues. What happens is during daytime the stomata, even if it is closed, it will not require any external CO2 because continually this CO2 will be produced from this oxaloacetate malate shuttle. So these crassulacean plants follow a unique pathway which is getting up during night time and sleeping during daytime, and that is the reason why they are exhibiting the reverse circadian rhythm. There are some benefits of this particular process. The benefit is because the stomata remain closed during daytime where the temperature is very high, it is a way by which the plant can evade loss of excess water by transpiration. And at the same time, in very desert condition, there may be 97% water lost during transpiration and the plant will die. So if they exhibit CAM process, the plant can survive in this extreme condition by keeping the stomata closed during daytime. Now CAM plants or crassulacean plants exhibiting the CAM process, they are of two types. They are called the obligate CAM plants, that is they only use the CAM which otherwise is not followed by any other process. And on the basis of the malic acid production, these plants may be strong CAM plants or weak CAM plants, and that may include the pineapples or other succulents. But there may be another type of plant which are C4 plant which may be succulent like Lucia, but there the CAM process is not mandatory. It is called inducible CAM, where there is a switching between C3, C4 or CAM process, and depending upon the environmental condition, when the condition become too stringent, the plant get into this particular process of CAM. Otherwise, it is following C4 or C3 processes. So it is called inducible CAM. If we compare the C3, C4 and CAM process, obviously all the plants exhibit C3 process. C4 process is exhibited by sugarcane, maize or other tropical grasses like panicum, whereas CAM process is exhibited by succulents like pineapple, agave, etc. In case of C3 plant, the carbon dioxide fixation is by the Rubisco enzyme, and the first formed photosynthetic product is the phosphoglyceric acid, whereas in case of C4 plant, the carbon dioxide fixation is by the PEP carboxylase, and the first formed photosynthetic product is C4 oxaloacetic acid. And in case of CAM plants, the process of carbon fixation is carried out also by PEP carboxylase, and the first formed product is malic acid. And actually I have said that in many ways C4 plant and CAM plant may be interconvertible because they are producing oxaloacetic acid and malic acid in tandem. But obviously C3 plants are most energy efficient method because it is followed by many plants, but C4 plant in some cases may be less, but in other ways where the carbon dioxide fixation is more, the stored food also increases. But the CAM plant, the productivity is not very high, but it is the issue of survivability because the plant has to survive in the extreme stringent condition. So they keep their stomata closed, they conserve water, yet they survive carrying out photosynthesis. And of course, we must say that the normal process of C3 photosynthesis loses excess water due to photorespiration. C4 plant is in between, they lose less water, but of course because the stomata are closed during daytime, minimum water is lost in case of CAM plant. There are some aquatic plant which also exhibit this CAM process, and this CAM photosynthesis is exhibited by ferns like Isoetus or other aquatic or submerged aquatic plant like Sagittaria and Vallisneria. Now it is very strange that why do they follow this nocturnal acid accumulation? This is because of the fact that in case of these aquatic CAM plants, loss of water is not the issue, but there is limited supply of CO2. So what we see is CO2 limitation also make the plant to get into this CAM process. So in this particular lecture, what we have discussed is different methods of carbon fixation other than the C3 cycle, that is the C1 process, the C2 process, the C4 process and the CAM process. I have also discussed the different plants where these processes are carried out, the mechanism by which these processes are carried out, and the utility or the importance of this process. And definitely because of these additional processes, plants can flourish in extreme environmental condition. Thank you.